Hello, welcome back. Here's our last lecture in the Chapter 10 series, wrapping up the chapter with discussion on antimicrobial drugs and resistance. So we've spent all of the other videos talking about the mechanisms of how antimicrobial drugs work to target those differences between prokaryotic and eukaryotic cells, fungi, and viruses as well. And then how do we test their efficacy? How do we know they work? And what are some of the ways we can get them into our bodies to do that work? And then what are the potential risks and side effects of that? This last mini lesson is probably the most important one as far as our current state of affairs are in the healthcare around the whole world with antibiotics. So remember at the beginning, I said pre-1900s, there was really no treatment methods for infectious diseases. You just kind of had to rely on your immune system and supportive therapy to whether are you going to fight off this infection, the virus or the bacteria, or not. And so infectious diseases were like the leading cause of death among children for sure, and mostly adults as well. Infections due to injury or just contagious communicable diseases like tuberculosis or influenza, those types of things. And then after antibiotics, that rate went way down, which is great. And we had vaccines for a lot of the viral illnesses as well, kind of came on the scene. But now the leading cause of death is not infectious diseases. The leading cause of death are lifestyle diseases like cancer and heart disease and diabetes and sedentary life issues and things like that. But we are on the verge of potentially going back to the pre-antibiotic times, which is really scary because most, day-to-day people don't rely on antibiotics a lot to survive. Every once in a while, you might get a sinus infection or urinary tract infection or something along those lines, and you may have only take antibiotics every here and there. But there are some people that would die if they didn't have antibiotics because surgeries or persistent infections, or they have to have a catheter or an IV line, or things that we kind of take for granted that we are able to do because we have successful antibiotics. But we are growing more and more bacteria in our world that can fight against these antibiotics. And then what do we do? Right? We won't have anything else to fight with. And then we're back to, well, it's our immune system. Let's see how well it works. So let's just talk a little bit about what that is, what resistance is, how did we get here? So first of all, I want to make clear antibiotic resistance is not a function of the drug. The drug does not cause resistance. Resistance is a naturally occurring thing that's part of the bacterial population. There were penicillin-resistant bacteria found within 10 years, if not even 5 years, of the widespread use of penicillin. So it's not something that's new. It's been around since we discovered penicillin and all the antibiotics. It's just a naturally occurring mutation. Some bacteria are going to be susceptible to it, some bacteria aren't. What's happening is the way that antibiotics work is they kill the bacteria that are susceptible, and that's the point. However, they don't kill the ones that are resistant, and that resistance happens naturally. And if we only kill the susceptible ones, who do we have left? The population is increasingly more and more of the resistant genotype that can then pass those traits off to its offspring, and a lot of times through this horizontal gene transfer we talked about back in chapter 7. So in this diagram, let's say we have a population of bacterial cells, right? Just plate them out. There's just regular old, get them from the dirt or get them from your nose or get them from your skin or whatever. Just going to plate them out on a plate that has the antibiotic in it. So of all the susceptible cells, we're going to kill off most of those. And you can see those are all kind of crossed off with the X's. And then what's left? Just a couple. Each one of those colonies may be representing just one cell. So 2 cells out of that original sample were naturally resistant to the antibiotic. Since everybody else is wiped out, they're like, yeehaw, I got all the nutrients. I have all this space. I have no competition. And so they just grow and grow and grow and grow because they don't have any thing to hold them in check. And so now if that's your population you're dealing with, that's what you have. So imagine this petri dish as your body. You have just your normal microbiota, or you have the pathogen that's causing an infection. You take your antibiotics, you kill off the susceptible ones, but if not all of them are susceptible, now all that's left are the resistant types. and it may have squashed your infection, but now maybe you have them on your skin. We have Staph aureus, Staphylococcus aureus. Some people might be carrying methicillin-resistant Staphylococcus aureus, which we call MRSA, on their skin. It's just part of their normal microbiota. It's not going to cause a problem unless there's maybe a cut or a scrape and it gets in there. But now we have that antibiotic-resistant bacteria just being a part of our normal microbiota, which can then, get rubbed off or sloughed off to somebody else. So this is where the resistance comes from. I'm going to post a video. I highly recommend you watch it. It's a Harvard, I'll just call it the Harvard E. coli video. But it's basically over 11 hours, I want to say, something along those lines, less than a day's time, I think. I'll have to rewatch it. Anyways, it's basically showing the progression of antibiotic resistance in kind of real time. They've sped it up a little bit, but it's showing you how bacteria can acquire resistance as they move through higher and higher concentrations of antibiotics. It's a really powerful video, so I urge you to watch that and kind of think about what you're seeing. And this is happening in that big petri dish you'll see in the video, but this is happening around the world. It's happening in bodies, it's happening in hospitals, it's happening in villages and communities. So this is happening. It's something that we're going to have to deal with. So let's take a look, just like we had mechanisms of action of how antibiotics work, we can take a look at mechanisms of resistance. How do bacteria, how are they resistant to these antibiotics? So I have a list of mechanisms of resistance in the notes. We'll go through those. The first one, which I have illustrated here, says the bacteria may produce an enzyme that destroys or deactivates the drug. So if you remember back in our inhibition of cell wall synthesis lecture, we had the beta-lactam ring, right? So here's our lactam ring, and this is what was incorporated into the peptidoglycan, and the bacteria couldn't grow their cell wall. Well, if that gets broken, so instead of being a square, it turns into more like a open box, it can no longer do its job. It is not effective at blocking those NAM and NAG subunits. So anything that can break that bond to change the shape of the beta-lactam ring would deactivate penicillin. And so there is an enzyme called beta-lactamase, right? There's an ASE ending that we talked about. Beta-lactamase enzymes can deactivate penicillin. And that's what this big blue blob is. And this is penicillin right here. So there's an active site in the big blue blob of beta-lactamase. Enzyme penicillin goes in there. It cuts that bond, releases an inactive penicillin, and then it can no longer work against the cell wall production. So any bacteria that have acquired the gene to transcribe and translate beta-lactamase are now resistant to penicillin. I'm going to post another video in the helpful links and videos on Blackboard to show you how beta-lactamase works. It's just a cool little animation showing you how some of these enzymes work. Some of these enzymes work to make these bacteria resistant. And I think that video might be talking about expanded spectrum beta-lactamases. So not only can they break down active penicillin, but all of the penicillin-like drugs that have the beta-lactam ring. So it's pretty powerful. So they can destroy and deactivate the drug. That's what we see here in this diagram. They can slow or prevent the entry of the drug. So if the drug has to get into the cell to function, they can maybe block that entry. And so if the drug doesn't even get into the cell, it cannot do what it needs to do to maybe block protein synthesis or cell wall production. Maybe you could alter the receptor of the drug so it cannot bind to its target. So the cell might be able to modify its own surface proteins to block the activity of the drug. For metabolic pathway inhibition, so we saw the PABA pathway using that particular enzyme that converts PABA into dihydrofolic acid. Bacteria can modify those pathways. So if they can find another enzyme to circuit, kind of reroute around that mimic, right? The sulfa drugs were the mimic of the PABA, then they can get their nucleic acids, and that drug then becomes useless if the bacteria has found a different alternate metabolic pathway, then the drug becomes obsolete. They can quickly pump the drug. They're called efflux pumps. The drug diffuses in and they can just pump it right back out before it even has a chance to do its job. And lastly, the bacteria can actually be part of what's called a biofilm. So hopefully you watched the Bonnie Basler video back in chapter 6 talking about biofilms. They do produce this protective layer of extracellular matrix and capsules and slime layers and fimbriae and flagella that the drug tries to get in and it can't. So if you have a biofilm, it makes it really hard for the drug to actually reach into the cells. So those are the mechanisms of resistance. So we had produce an enzyme like we see with the beta-lactamase, slow or prevent the entry of the drug, alter the receptor for the drug, alter the metabolic pathways of the drug, pump the drug out, or become part of a biofilm. It's kind of scary. I mean, these are the things, but these are naturally occurring reactions, and they are selected for. Antibiotics select for the resistant strains. They kill the susceptible strains, and all that's left are the ones that are resistant, and they are the ones who get to spread their genes via that horizontal gene transfer and to their offspring. Okay, so what can we do? So I have some things to mechanisms on how to slow resistance through our communities and globally. So first of all, if you are taking antibiotic, maintain the high levels of drug for a long time. So if you are in that oral medication administration route, take your medicines as directed for the whole time. Don't stop taking them after a couple days once your symptoms improve. Use a combination of drugs. So maybe if you have a severe infection, talk to your doctor about, well, can I have one that targets cell wall production and one that targets protein synthesis? So if there's resistance in one, maybe they'll get hit with the other, or some kind of combination of synergism between two mechanisms of action against the pathogen. Don't overuse antibiotics. You can't treat a viral illness with an antibiotic. And that's hard for some patients. They have signs and symptoms. They want to go to the doctor. They want to get a pill. And the doctor's like, meh, here you go, some penicillin. But it's not going to do you any good because you have a rhinovirus, cold, or influenza. So that's kind of the people part of it. And then there's quite a bit of antibiotics used in agriculture. So if you want to take a guess, a percentage of all of the antibiotics that's used in the United States, how much do you think is used in agriculture? 10%? 20%, 50%, 80%, 80%. About 80% of all antibiotics in the country, in the United States, are used in agriculture. It's not for humans, it's for agriculture and farming. Why? Why give a cow antibiotics? Well, think about where cows and sheep and chickens and pigs and stuff live in farming practices. Do they get to be socially distant? No, they're all crammed together. So if one cow's sick, all the cows are going to be sick. And if you're a farmer, you don't want sick cows. So let's give some low-level pre-therapeutic antibiotics. So if there is an infection, they're going to be able to fight it off. Well, the problem is, yeah, you might be fighting off some illnesses and keeping healthy cows, but you're selecting for all the resistant strains. And then the resistant strains get into the environment. They're washed in the poop and the irrigation water and down into the streams. And so now you have all these resistance genes mixing in the environment. So limit antibiotic use. So that's why it's good if you have a choice. If you can express that choice, try to purchase items that are grown without antibiotics. That's your voice as a consumer to say, I don't want my food that I'm eating to be treated with antibiotics. And then lastly, try to develop new drugs, which is really hard in our pharmaceutical industry. There's not a lot of incentive to develop a drug that the patient is only going to use for a week, right? If you are a pharmaceutical company wanting to make money, you want to make a drug that patients might need for a long time. So a lot of the drugs that you see, like on commercials and stuff, which I will step out of my instructor role and put a personal opinion in here, is I don't think it's right that our pharmaceutical companies can advertise to customers straight through TV. That's my personal opinion. That's Corey's opinion. That is not RCC's opinion. Back into the teaching mode. Okay, so a lot of the commercials that you do see are for chronic conditions that the patient, and that's great. I think I'm not trying to slam that there's medications for people that need medications. I think that's a good thing, but that's where the money is for these pharmaceutical companies. Antibiotics people only need for a short period of time. So why would you want to spend a lot of money making a drug that is not going to be needed for long periods of time? So we need to, as a society, as a government, to incentivize new developments for drugs, make it worthwhile for these scientists and pharmaceutical companies to want to develop better antimicrobials to fight off some of these bacteria that we don't have anything else to fight them with. Anti-quorum sensing, so that was kind of a focus of the Bonnie Basler talk, is here is this thing we're recognizing, these chemicals we're recognizing the bacteria have, could we target that to stop their replication? And then I'm going to talk a little bit in the next slide or so of what's called bacteriophage therapy, which actually was started before antibiotics were discovered. So we'll talk about that. So these are some ways to slow resistance. Keep taking your meds, maybe combine them a couple times with a couple other ones if you're having a severe infection. Limit antibiotic use, it's way overprescribed, and develop new drugs, incentivize that. So take a look at some of this, some results from some of these bacteriophage therapies. So you remember bacteriophages? No, you don't, because we haven't got to bacteriophages yet. Chapter 13, that's our next chapter we're going to get to. But we saw them a little bit in transduction. So bacteriophages are viruses that infect bacteria and they kill them, right? In that lytic cycle. We haven't covered that yet. My brain's all mixed up. So in the lytic cycle of bacteriophage replication, when a bacteria is infected with a bacteriophage, it kills the bacteria. And bacteriophages, these viruses, do not infect human cells. They do not infect eukaryotic cells. Their targets are very specific to the bacteria. So a bacteriophage that could target this is Staph aureus, would not affect any of your E. coli in your guts. And so that side effect of normal microbiota is lessened. Disrupting that normal microbiota would not be a factor for these bacteriophage therapies. So here I have kind of this person had a skin infection with Staphylococcus aureus, which is probably MRSA, methicillin-resistant Staphylococcus aureus. And so you can see in November, it's kind of this opened ulcerous wound. And then this phage bioderm is a patch that was impregnated with multiple different strains of bacteriophages that target the pathogen S. aureus. And you can see over November, December, January, February, March, about 4 months time, kind of with this, I don't know what the treatment plan was, but I'm sure exposure to this wound with this patch particularly, you can see a pretty significant healing of that tissue in just 4 months, where other types of antibiotics may not have worked, but the bacteriophage therapy did. The history of some of this is that back in the turn of the century, again, early 1900s, a lot of countries, even the United States, was working with bacteriophage therapy and seeing if we could find ways to treat some of those infectious diseases. But what happened in the United States is Alexander Fleming's discovery, penicillin, sulfonamide, and they're like, yay, these miracle antibiotics, and kind of put bacteriophage on the back burner. And it's just now kind of coming back up to the forefront because of this situation of antibiotic resistance that we are dealing with now. So I'll let you read through some of these tables, but there's advantages, but there's also going to be some disadvantages to this type of treatment. I wanted to share with you some research. I had to do a research project on antibiotic resistance. So this was just one slide of a study that was done to look at vancomycin resistant Enterococcus faecalis. So this is the little critter, the bacteria that causes these deadly infections. Vancomycin is a type of similar to penicillin, so it affects cell wall synthesis. So bacteremic mice, so they have a bacterial infection in their bloodstream with this VRE, so the vancomycin resistant strain. They have a particular bacteriophage, this guy over here, Enterococcus ENB6 bacteriophage. So this bacteriophage targets this bacteria and kills it, only the bacteria. And then here's your little critter, the mousy mouse, that was the subject of this particular study. So what they did was they mixed the Enterococcus faecalis and the bacteriophage. I don't know if they did it in the same container beforehand, or they made the mouse sick with the bacteria and then they gave it the virus. Either way. So what they did was they were looking for the concentration of virus that would be successful in the survival rates of the mouse. So here we had 3 times 10 to the 9th, 8th, 6th, 4, and 0 virus. Okay, so that's kind of what those numbers mean. So 3 times 10 to the ninth, I think those might be our control. So they were not infected by the bacteria, but they were given the bacteriophage. So 3 times 10 to the ninth with the bacteria and the bacteriophage, we can see after day 1, 2, 5, and 6, we had 5 mice start, and we have 5 mice at the end. So that's a really good concentration, but could we go less? So 3 times 10 to the negative 8, we start off with 5, we ended up with 5. So that's great. And these are just measures of health. So your y-axis is the state of health. So 3 times 10 to the 8th is a good concentration of bacteriophage. Then we had 3 times 10 to the negative 6. Started off with 5 mice. 1 was really healthy, 4 of them not so much. And then by the time we get to day 6, 3 of them kind of all spruced up and 2 of them died. So we're kind of on that bubble of a good concentration, but not for so much. And then we had 3 times 10 to the 4, only 2 mice survived, and 0 bacteriophage, 0 mice died. So what that means is all of the mice that were infected with the bacteria with no treatment died. 13 of the highest concentrations of mice that were infected with the bacteria and the bacteriophage survived. So this was evidence to show that bacteriophage therapy works for this particular case. And then with that, they would go forth and try to do more trials to see how well it worked, maybe in other systems. Oh my gosh, my cat just scared. Okay, one more slide. So this last slide I'm bringing up, this is just a screenshot from the CDC's website. They have reports. They did one in 2013, and I think this one was 2019. So these are what we call CDC's biggest threats. So they have collected data from all over the country to say, what are the biggest problems? Which organisms are our biggest threats that we are running out of ways to treat? And so I just put a snapshot of one of the more urgent ones. I think there's like 4 or 5 urgents, and then they have serious, and then they have threatening or something like that. I don't know what the yellow is. So this is called carbapenem resistant enterobacteriaceae, big mouthful, CREs is what they're called. So these guys are super scary. These are like crazy scary. So here's just some statistics: about 13,000 cases in 2017, about 1,100 deaths, and it costs about $130 million in just dealing with this particular infection, not the condition that led to this, but this particular infection, usually a healthcare acquired infection that they get it while they're in the hospital, not that they got it and then go to the hospital. So we really have, here it says, a family of bacteria that are resistant to nearly all antibiotics. This is that pre-1900s state that you are in. So if you get a catheter infection or a urinary tract infection or pneumonia or a sinus infection by one of these CRE bacteria, you have no recourse. There's no antibiotic that would work against these guys because carbapenem was the last resort antibiotic. And now there's bacteria that are resistant to our last resort antibiotic, and this is them. And this includes things like E. coli, Klebsiella pneumonia, I think Pseudomonas. No, Pseudomonas aeruginosa is not in there. There's a couple other ones. They're not giving species names, but I know Klebsiella and E. coli are part of this Enterobacteriaceae group. And you can just see estimated cases of CRE in hospitalized patients from 2012, 11,000 up to about 13,000 in 2017 is the year that's kind of blocked by my picture. It's a lot, and it's going up. It's not going down. So what can we do? What can we as a society, as a research country, do to try to come up with solutions for these CREs? If you wanted to look at more biggest threats out there, it's kind of interesting. You can just kind of click to their website. This came from the report. There's like a 150-page PDF that you can kind of scan through. It's actually pretty well written. So I give the URL here if you wanted to follow that and see some of the other urgent, serious threats that you may be seeing if you do work in the healthcare field. All right, that is it. That wraps up our Chapter 10 discussion on antibiotics. Okay, I will see you next time. Bye.